Hi, good afternoon, everyone. I'm Danielle. I'm a PhD student in Professor Karen Avram's lab, and I'm very excited to be chairing the final session of the day. So I'd like to invite to the stage our first speaker, Professor Anat Ben-Zvi from Ben-Gurion University of the Negev, who will be talking about the landscape of molecular chaperones is modulated in aging.
כן. זה את הסופר שלך הם צריכים לשנות?
Of sitting in Zoom room and being by ourselves. This is a bright change. I want to talk to you today about how we look at the quality control system and its changes in age and age-dependent diseases and how those contribute to age-dependent diseases. I want to start with, no, just a second. Oh, great. No, it's weird. Yeah, it's like, this is, move to the next one. Okay, so this one is good. So what's wrong with this? Oh, something is weird here. Okay, we'll try to work around this. Many diseases are actually manifesting clinically in one or specific number of cells. It's very common in many diseases and very common when we look at age-associated diseases. Neurodegenerative diseases is a great example of that. We see neurodegeneration that not only affect just neurons, but sometimes even subtype of neuron are affected. In an examination of Mendelian disease, where it's easier to look at it specifically, I hope you can see partially that most of the diseases were actually impacting one or several tissues. On the other hand, when we look at the expression of the genes that is causal for the disease, that is not limited to one or few tissues, but in fact, for most of the diseases, it's genes that are ubiquitously expressing. And so this is basically saying that although we have a gene that is expressed in many different tissues, we see the disease presented mostly in one or several tissues. And there are many mechanistical explanations that are looking into this. Some are as simple as expression level. If we have a tissue, for example, that is expressing high level of a particular gene, then obviously if that gene is affected, this tissue is most likely to be affected. But there's also other explanation like partners that are in one tissue or another, paralogs, or a redundancy of function. And as I said, what I'm interested in is quality control mechanism, and specifically what I wanted to ask is whether or not quality control system can play a role in tissue specificity of disease. So for those of you who are not familiar in quality control machineries, they are basically divided into two arms. One is composed mostly of chaperone, which are proteins that assist other proteins. They are able to specifically monitor and recognize proteins that are misfolded or have some kind of problem or damage, and they help them to recover them and restore their function. The other arm of the quality control system is the degradation machinery, mostly the autophagy and the proteasome system. And this arm also can recognize damaged proteins, but send them for degradation, basically remove them from the cell. The two arms obviously cooperate and talk to each other, and together they form the quality control machinery. Today I'm going to focus mostly on the chaperone system. So asking if the chaperone quality control can impact the specificity of hereditary diseases is basically asking a very fundamental question of how quality control systems are organized in multicellular organisms. And this is because we can envision two extremes. One, when we sort of say one size fits all, where we have a master regulator that controls the expression of chaperone in any given tissue, and then we will have a certain machineries that are producing those cells, and they do the job, and it doesn't really matter if you're a muscle cell or a brain cell, this is the function you're going to have. And then on the other hand, we can envision a tailored system where if you are a muscle, you're going to get the quality control that a muscle requires, whereas if you are a neuron, you're going to get the quality control that the neuron requires to do the function. And so what we wanted to ask, if the chaperone network differ between cells, if they do, why is that? An assumption will be that it's to fit the needs of the folding environment, but it's an assumption that needs to be examined. And then, of course, whereas we sit and speak, is whether or not this is impacting aging and age-dependent diseases. So to start with, we ask whether or not the chaperone are expressed in multiple tissues or expressed singularly in specific tissues. And what you can see here is that most chaperones are indeed expressed in most tissues, so they are ubiquitously expressed. And in fact, they're significantly more ubiquitously expressed than most of the protein coding genes. So this is a very highly expressed, very broad expressed family of proteins. But being broadly expressed, as I noted before, is not necessarily means that you're equally expressed in all cells. You can have differences in expression. And to look at that, we took advantage of the GTEx consortium data set, which gives you, for each tissue, many different samples that you can then look at the changes within samples, basically between individuals, and then between tissues, and see whether or not changes in expression are significant and how so. And what we did with this data set, among other things, is we did a cluster analysis to look at whether or not the expression profile of chaperone is organized in some manner. And what we found is that, indeed, similar tissues actually show similar expression patterns which cluster together. So muscle and heart muscle seems to show specific chaperones that are going up and specific chaperones that are going down that are different from what you would see in different brain area or skin and so on. This suggests that the proteome that is being expressed by those cells have some impact on the chaperone or quality control that those cells express. This is in agreement with things that we and others noted in the field, where you can see that as part of differentiation process, there are changes in chaperone expression and quality control component in general, whereas some chaperones are going down, and some chaperones are going up, and some are unchanged. And in fact, if you assume that quality control systems are great because they fix things and so on, this is not necessarily so, because you can sometimes overexpress a protein and get damage. And if you take a protein that's normally downregulated during differentiation and force it up, you can actually get disrupt differentiation and other processes in those cells. So it seems that yes, indeed, there is tissue changes, and those tissue changes could be affecting or addressing the cell need. This is also apparent if we look specifically at disease. So if we looked at disease of chaperones, so chaperonopathies, chaperones that have mutations, there are 10 diseases that affect muscle. Of those 10, 7 are actually proteins that are upregulated in this group. The other disease actually don't affect just muscle, they affect multiple tissues. If you look at the rest of those chaperones that are upregulated, almost for all of them you can find association to protein folding in muscle, and you can find association to disease, not causal association, but other linking association. You can also see that this is not just a functional impact, but also a conserved one, as the chaperones that are upregulated in human muscle are also conserved in mouse, And in fact, they are also conserved in C. elegans. So most of those chaperones that are upregulated in human muscle are also highly expressed in C. elegans muscle, which is quite a different. So this, in all, says that we have a conserved functional regulation that ends up in having a specific patterns of quality control that is expressed in a given tissue. We then zoom in a little bit into what a type of changes do we see and what exactly, or how exactly is this system organized. And in general, we could see two phases of organization. One, which is the variable chaperones that their expression changes across all various tissues, and then core chaperones that don't even change twofold in any tissue that we tested. So those are stable in their expression across the different tissues. And when you look at these core chaperones, what you see is that they share basic functional, cellular functional roles. Let's say chaperones that sit on the ribosome and help a protein get out and fold, or chaperones that help translocation into the mitochondria or into ER, or folding and degradation in the ER. Things that are really basic to what a cell needs in quality control, whereas the variable chaperones were much more distinct in the terms of the function that they were assigned to. What was more interesting is that core diseases, most of them, or core chaperones, most of them did not, we could not find mutation that caused diseases in those chaperones. For the most part, there were only 3 that were associated, whereas in the variable chaperone, almost all the diseases that are causal by chaperone were in variable chaperones. And this, you can assume, is because the core are not important, but in fact, if you knock down a core chaperone and look at the viability of the cell, core chaperone has much more impact on cell viability than the variable chaperones. So this is suggesting that the variable chaperones are allowing or are more unique or behave differently in different cells, and so you can impact them without having embryo lethality and so on. And this is also going back to what we started this talk on, looking at the tissue specificity. If the variability or the chaperones that are variable are affected, the effect will be dependent on the profile in different tissues and their importance in different tissues. And indeed, when you look at that, what you see is that most chaperones that are associated with disease manifest clinically in a few tissues, but they are actually expressed in all the tissues, similar to what we saw in the general, let's say, the general Mendelian group of diseases. Chaperonopathies stick to this rule. I hope the next slide looks okay. It doesn't really. What I wanted to try to convey here is that we can look at the chaperones as a system, but we can also look at the network and their interaction or correlation. And here what we've tried to see is whether chaperones that go up or go up together in different tissues, or whether they're disconnected. So in one tissue they will go up, and in another tissue there will be one go up, one go down, and so on. And this is an example of one set of chaperones, the CCT chaperones that builds one complex. And I hope you can see that when you look at the core chaperones, what you get is that for the most part, it's heavily red, meaning that they are highly correlated. So one subunit goes up, the other subunits are also highly expressed. But then when you look at more variable set of CCT subunit, you see anti-correlation or you see no correlation in their expression, suggesting that this organization also impact, I'm missing one slide here, missing one figure, correlated to this behavior. And I had a part here that talks about quantification and basically show that if you look at the pair behavior, what you see is that core pairs tend to be across many different tissue correlated, whereas mixed pairs seems to be in one tissue they are correlated, in another they're not, and so their correlation is not very strong. And what this suggests is basically that even for the core chaperone, but most for sure for the variable chaperones, their interaction and partners are going to change between tissue. So if I have a chaperone in one tissue, it will interact with a set of proteins, but in the different tissues, it will interact with a different set of chaperones, and therefore create a different network that will have a different functionality and different capacity. Okay. I don't know what's going on with those slides, but okay, so what I wanted to stress here is that we can look at how those modules are created in development and then how they are maintained in aging. And one of the things that we noted is that genes that seems to be expressed in a particular tissue are also correlated in the adult tissue. And so this signature is, as I noted before, start to form when differentiation and maintained through development and adulthood. But what's more important and I guess interesting to this crowd is what happened in aging. And here what we did is we took another data set that looks at the aging brain and looked at chaperones in brain tissues that their expression reduced over time. So basically we're looking at chaperones for which the expression either reduce or increase with age, or basically change with age. And we ask whether those chaperones are similar to the chaperones we see in the unique signature of the adult brain. And for the most part, we had an overlap of 265 pairs, which seems to be, it is significant, seems to be small, but if you look at this particular set, what you see is that first, most of them are associated in one tissue, so they are unique to brain. And second is that the 258 of them, which is almost all of them, are actually belonging to the group of chaperones that are repressed in aging, meaning that those chaperones, expression goes down. Those are the chaperones that are also associated with what a chaperone brain needs, or what the quality control in brain needs. So the same chaperones that brain upregulate or tissue, neuronal tissue upregulate in order to protect and have their quality control system, those are the same chaperones that go down with aging. And you can see this particularly here, the core chaperones and the specific variable chaperones are reduced, whereas the other variable chaperones that are just changing, those are remain unchanged with age. Now I'm just praying that the last slide is okay. Okay, so this is also true in a Alzheimer disease patient. So if we can compare them to age-matched individual, what we see that the core and the upregulated, the signature chaperones that are expressed in brain tissue are reducing further in Alzheimer brain patient, whereas other variable chaperones that are not specifically unique or highly expressed in neurons are the same. So if I want to summarize everything I told you, and I regret some of the slides being not exactly what they were supposed to, what I showed you is that the chaperone system in multicellular organism, and specifically in human, is composed of core and variable chaperones that basically produce tailored quality control system that is geared towards interacting or answering the challenging of a particular tissue. So if you're a muscle and you need to build a sarcomere, you will have the chaperones that needs to do that, the basic chaperones, but also variable chaperones that are doing this function, whereas if you're a neuron and you're secreting vesicles and you need a lot of chaperones that maintain that, this is the type of quality control you will have. And this quality control system is affecting the capacity of the cell, affecting the functionality, and has strong impact on disease in general and age-dependent specific diseases. And basically the landscape of molecular chaperone is critical when we look at aging and age-dependent diseases. With that, I need to thank the people that did the work. First and foremost, I have to acknowledge my really true collaborator in this project, Esti Yegerloton, which is a system biologist, Serena Kara, that shared with us some of the muscle analysis in this project, and the two really brilliant students that did most of the work, which is Neta Shemesh from my lab and Juman Jubran from Esti lab. And with that, and Ministry of Science that gave us the funding. And thank you all for your attention.